Hello everyone, my name is Dylan Hamilton and welcome back to C.A.R. Entertainment. Today we are going to be talking about sources of genetic variation as a continuation of our A22 biology revision videos, guys. This is just going to be about genetic variation and how it will occur in humans and different mammals and species essentially as a whole. So we'll go straight into it and talk about our first source, which is meiosis 1. If you did AS biology, which I'm sure you would have done if you're in the A2 part, then you will know all about meiosis. And so there's 3 different forms of meiosis 1 that will act as a source of genetic variation. The first one is independent assortment, because when one chromosome from each homologous pair will enter a gamete, which of two chromosomes will enter a gamete will be dependent on how the homologous pair line up at the start of metaphase 1. You then have crossing over, which is in prophase 1, which facilitates the exchange of genetic material between the homologous chromosomes. And finally, cross-fertilization, which is whenever the male will produce large numbers in gametes, each with different chromosome and allele arrangements. The second source is mutations, because mutations are restricted to a single gene and they will affect one base called a point mutation. Substitution is an example where one base was replaced by another, and this point mutation will involve substitution, which is limited to one amino acid. If substitution takes place in the third base of the triplet, then it may be no effect due to the degenerate nature and the mutation is neutral. In sickle cell anemia, the normal cell DNA-based triplet is CTT, and this codes for glutamate, but the mutated sickle cell anemia has the allele of allele DNA sequence known as C.A.T, so you replace that middle T with an A, and this codes for valine, and therefore you get sickle cell anemia. Deletion mutations is a point mutation that has a consequence of significant cause, as it will cause a frame shift mutation where DNA will be read and freeze, as we know, but by deleting one base change, then the base triplet along the rest of the gene will be different. And this means all amino acids after the point of the mutation will be changed, or the protein will be shortened if the stop codon is produced earlier. In terms of chromosome mutations, this will involve a change to large sections of DNA or even whole chromosomes. The mutation to Down syndrome is by one parental homologous pair failing to segregate properly, resulting in gametes produced with 24 chromosomes. If the affected gamete involved in fertilization in a zygote, then there's 47 chromosomes produced, and this results in Down syndrome. Down syndrome is aneuploidy, which is a situation in which one chromosome pair will fail to segregate. Aneuploidy will lead to affected individuals having a missing chromosome. If all homologous chromosome pairs fail to segregate, polyploidy results. Polyploidy can result with 3 sets of chromosomes known as triploid, or 4 sets known as tetraploid. The cause of mutations is that they can occur spontaneously, and the rate is accelerated if you're exposed to environmental stimuli. UV light and mutations give rise to uncontrolled cell divisions leading to skin cancer. X-rays and gamma rays can also increase mutation rates, and a variety of chemicals such as caffeine, tobacco tar, and pesticides also cause mutations. Thanks for watching this video. I've been Dylan Hamilton from C.A.R. Entertainment, and I will see you next time.